Hello, everybody. Welcome to the Mid-Atlantic Micro-Nano Alliance YouTube Live webinar series. I'm Heidi Leonard, and I'm here today with Professor Ryan Sokol from University of Maryland College Park, co-president. So Ryan, who do we have the pleasure of hosting today?
Hi, Heidi. Hi, everyone as well. So we're here with Professor Daniel Cohen from Princeton University. And so Daniel actually did his undergraduate training in mechanical engineering at Princeton a long time ago. And then he did his PhD at, not that long, I don't know, it was long for me at least.
This is a long time.
And Professor Cohen did his PhD at UC Berkeley in bioengineering in an electrical engineering laboratory within the Berkeley Sensor and Actuator Center. So, for a bit of time. And he also did a postdoc at Stanford University in cell biology. He now runs a lab at Princeton University, combining all of these things for pleasure and profit. And so now I'll introduce Professor Daniel Cohen.
Not so much the profit. Yeah, thanks. I'm Daniel. It's good to be here. And I always like talking to interdisciplinary audiences, and I got to get some engineering fixed in soon because I've been talking to a lot of cell biologists. So I will talk a little bit about what we do, give a little bit of biological background, and then ask for some advice on some of the problems we're having. So here we go, Zoom. I swore I was never going to do cell culture again after undergrad. Didn't like it. I didn't like the lack of engineering, and now it's all we do. So what converted me back was when I did an experiment with kidney tissue at a training workshop one summer in the middle of my PhD. And I transitioned from doing dinosaur biomechanics and nanotube electronics to tissue biology after this experiment that you see on the left. So it's a kidney tissue that I cut a plus sign into and then stayed up all night watching it heal itself. And it's really beautiful. It doesn't really happen the same way twice, but it always happens. Those are thousands of cells all coordinating their motion to heal the injury. The blue dots are the nuclei of the cells. The red is protein where they're holding on to each other. And as an engineer looking at this, it gets really interesting because you have this material made up of all these sort of microscopic agents that are all working together to sort of solve a problem. And you don't have to do anything to get them to do this. They will always do this. And this concept of sort of tissues as communities relates back to an old 1800s idea from Rudolf Virchow, who started physiology or pathology as a field. And he says that tissues and organisms are like societies of cells with tiny, well-ordered states and all of the accessories and officials, underling servants, in other words, the bureaucracy of a nation state or a city or some sort of complex social organization. So it really is fair to think about these large collective systems as being communities. They have waste collection and mass transport through blood vessels. There's communication lines like nerves and neurons and motors and muscles, and then there's structure like soft tissues, collagens, bones. So all of these things work together to allow multicellular things to function. And they do it without our input. But if you're an engineer, the question you're asking is, well, if I poke it, what can I do to it? And so that gets exciting because then we think about how we can take these systems that already have these embedded processes that are really sophisticated and make them work better. Can we use microengineering techniques to speed up how a tissue heals, or to grow a tissue in a new way, or reduce scarring? And those are questions that require kind of taking into account the collective behaviors and coming up with engineering approaches to manipulate those. And there's all sorts of fun tools you get to use along the way. So we're going to very quickly go over three basic areas. Basic of collective cell behaviors. And so you get a sense, if you're not familiar with it already, of cells as materials and kind of just how you can work with them. And then two examples of an outside-in type thing, like sheep herding of cells, and an inside-out type thing based on micro devices that mimic cells. So starting with some behaviors of just what collective behavior is, how we think about it in tissues, and kind of how you mess with tissues. Hopefully you've had a chance to see bird flocking before. It's really pretty, happens all the time. We understand a lot of the dynamics about this. We understand how the birds kind of keep track of a certain number of neighbors. It's a pretty neat process. And again, nobody tells these what to do. And this type of process can happen in any sort of system that has a sufficient number of agents with particular coupling or interactions between them. And so that we're on the same page, if we look at cell systems that do this, we'll build up from single cells to some of the behaviors they do. So this is a single kidney cell. Nuclei here is essentially running code that's environmentally responsive. Sensors in the cell detect changes in the world around it and can program or induce changes to the program. So the cell might contract or produce new proteins or divide in response to stimuli. It's got motor proteins around it, these green motor filaments that can contract and generate tension, and it attaches to the world around it through yellow little feet here. And that's how it moves around on a Petri dish. If you get two cells together, that's where the magic starts to happen from a materials perspective. Suddenly, now you have a composite of two cells that are holding hands along this red boundary, which is in 3D. It's where they're literally attached to each other through special adhesion proteins that are unique to each given type of cell. When they do this, you can see they're perfectly balanced now. They have their feet all built up in these corners and they're kind of in static equilibrium. Once you get enough of these together, you get symmetry breaking. You suddenly get a tissue with instabilities that starts to fluctuate. And hopefully this really nice movie is showing you these patterns of undulations and flow within a tissue. And now you have something that over time will slowly grow and become a mature tissue and start to get more and more kind of stationary, but can be reactivated by injuring it or doing other manipulations. So you have a living sheet of material and you can grow these to arbitrary sizes. Keep in mind that mass is not conserved here. So anytime you see a blue dot flash white, it's dividing and becomes two. That's coming out of the liquid around the cell. It pulls raw ingredients out and makes more of it. So it's a really interesting kind of smart material to play with. And the last sort of rule set you need to just think about how these systems behave together is that these complex processes like healing derive from local interactions amongst the individuals. Like any crowd-based process, you need some coupling from individuals within the group to say their nearest neighbors. And if everybody has a certain set of rules they're applying locally, those can create propagations that give rise to more complicated global processes. And these processes are greater than the sum of their parts. If you take a person out of a cocktail party, they're going to behave very differently in that non-social context. Cocktail parties are things that adults used to be able to go to before COVID, in case you're too young or you've never had a chance to do that. And so taking a cell or a bird out of a flock is the same sort of thing. If you remove something from a social context and then you analyze everything about that individual, you're not going to know how it would have behaved in the group. So you have to think about these things as the whole system. And lastly, again, nobody tells these things what to do. Boundary conditions change, perturbations play out, and the system responds in a sort of statistically predictable way. So how do you work with these things? Just in case you haven't had a chance to play with them before. They really are pretty versatile. This is a patterned tissue that's growing into a shape over, say, 24 hours, and we can micro-pattern that. These are blood vessel cells zipping along micro-patterned roadways. And the concepts to just keep in mind are, you can micromold tissues, you can put them on textured substrates or patterned substrates that change their behaviors, you can bioprint them, you can integrate them with advanced nanomaterials like electrospun nanofiber scaffolds, or basically anything that has high surface area that you could functionalize with something like collagen so the cells stick. And as a material themselves, casting, molding, spinning, self-growing, they can even produce their own materials. There are some companies that use cells to produce structured nanomaterials by having them template down special types of fiber matrices and things. And again, mass isn't conserved. You have a system that's constantly capable of expanding. So that's the medium that we like to play with. And now we can introduce some of the reasons why that's useful. So why would you want to control these things at the collective level, especially in a Petri dish? And there are a number of basic explanations for that, but this is the basis for engineering tissues outside the body. Things that we do in the dish, we can transfer into sort of translational research to try to better understand how to heal injuries and things like that. But everything that we look at in our lab, at least, is based on these behaviors of large numbers of cells. And we have this idea that if we understand those basic collective behaviors and find the equivalent of a sheepdog to poke them in different ways, we can make them work for us to do something useful. So an example that we'll talk about first is this kind of way of interacting with smart materials. And if we take a very quick example of what that looks like in a simple case, here's a single tissue on the left here, and here's how it grows over time. And we just published a study of how large tissues grow. This is a tissue over 1 millimeter in size. Most studies are on micro scale, and we found a lot of weird things that happen. Punchline, doesn't matter what happens on the inside, this crazy spinning vortex doesn't affect the outside. These tissues grow according to a fixed rate, and that makes them extremely easy to predict. This is skin or kidney tissue here. So then we said, well, what if we put two of them together? They do exactly the same thing, but when they meet, they pin. And the only difference here is color, so it's the same type of tissue. And turns out pinned boundaries are actually pretty common in tissue collisions, and it's one of the reasons why we can have organs that are separated from each other by compartmentalization. It's kind of important. And we can start to use that as an engineering trick. So we say, okay, this collective has two things that it does for us. It expands on its own in a completely predictable way based on any given shape. And when it meets another thing, it kind of stops moving and follows the boundary. So now you have living boundaries that are growing and changing over time. And you can get fancy and say, well, what if we just made more fancy patterns? What if we patterned 5 of these tissues together and just let them grow? So again, these are in a dish on a collagen surface. So it's a 2D culture right now. And we simulated it using a tool based on the data from these other 2 studies and predicted that we would be able to make patterns like this. So you're thinking about sort of multi-material composites now. And we tested that with actual tissues, and hopefully this video plays. You have 2 green tissues in the middle of some purple tissues. And as they grow out, they form those predictable patterns. And that's kind of neat because, again, we didn't really have to do much, and we got these fancy outputs. And so we turned that into a tool that we're calling TissEllate, which is a way of tessellating little tissues into predictable patterns. So I challenged a grad student, Matt Heinrich, to make something kind of like an Escher drawing with tissue. So here's an array of cubes with 3 different colors, white, black, and gray. And we simulated it and found that if we had little tissues, these are about 1 millimeter long, of kidney tissue, patterned down in this initial pattern, and we just let them grow over a day on their own, doing whatever they normally do, they spontaneously form a stable structure like this with clean boundaries, multiple different types of tissues scattered in predictable locations. And you can imagine doing this with different types of tissues or tissues running different sort of genetic expression patterns or producing different chemicals and materials in different locations. And it's a really nice demonstration of how you can kind of synergize with existing collective behaviors when you're engineering them. And as long as you have the ability to kind of micro pattern these things, which is cheap and easy, you can produce these pretty complicated things without too much effort. So you don't need something like bioprinting to produce certain types of really complicated interlocked tissue structures. So that was just an introduction, so you have a sense of what the medium that we work with can do and how it grows and interacts. So the next version of this then is like, why would we want to control these? What other things could we do? What you saw was us kind of letting them shape themselves based on our initial conditions. Now we want to say, well, if you imagine them as a bunch of little robots all interacting with each other as a big sort of soft material moving around, can we steer it? And that's kind of fun because we get to think about a different problem, like sheep herding. So what could we do if we could steer cells like sheep? Well, let's just refresh ourselves of what sheep herding looks like. This is one of my favorite movies. It's drone footage of sheep herding. This is one of our oldest collective technologies or swarm engineering tools. It's maybe a 7,500-year-old set of techniques for herding animals. And you can see the dogs as little black dots moving around doing their perturbation that causes the flock to change. We don't know how this works. There are lots of papers on theories of sheep herding. There are papers published each year on heuristic models for how the dogs and the sheep interact. And the dog certainly doesn't know, and the shepherd can do amazing things without any advanced model. What's really nice about collective engineering is, again, if you can rely on the existing behavior, all you need is a good perturbation and the ability to do feedback to watch and adjust things and kind of let the thing shape itself. So could we do that with cells? And if we could, what would that buy us? And the answers that we've thought about so far, at least, are you could sculpt tissues. If you were growing organs in a dish, you could sculpt them in programmable ways. You could make a wound that's already going to heal itself, maybe heal itself faster, or maybe heal slower, but reduce scar formation. You can program general motion in different types of research contexts where you need to move cells around, and you can develop better models of just general collective cell behaviors if you have tools that let you turn them on and off. So it could be a really neat biomedical tool. The problem is you have a beautiful system like this with sheep, and you got to turn it into something that works with this on the right here. So how do you go from this image or this movie to this clean type of control? And the answer is that we made a problem a little bit more constrained, but also harder to try to come up with a new set of tools. So the first thing is we said it needs to be interactive control. It's great that you can micro pattern little grooves and tunnels and things that cells will move through and basically stay along the pattern you've printed for them, for instance. But you can't change that over time. So we want something dynamically programmable. We want it to not kill the cells after a long period of time. We want us to be able to turn the stimulus on and then turn it off and have everything go back to normal. And we want it to be applicable broadly. If you have to genetically modify your tissue to have all sorts of special receptors and markers and things, that's not as useful. So we asked, what could we do just with native tissue? And we're going to see how that works. And there's been a lot of work in this field in general of trying to control cell behaviors with things like optogenetics, which is an extremely powerful and cool technique where you use light to manipulate genetic activities. But that also requires special modifications to cells and can limit some of the behaviors you can control. So there isn't one perfect solution. What we're thinking about is kind of an outside-in, poke the tissue, see what happens, and program its motion. And you can choose different types of sheepdogs. The obvious choice might be chemical cues, because everyone knows that cells kind of exist in this wet squishing environment and follow chemical signals. That is true for certain types of cells that do a very good job of it, but many cells don't follow these signals particularly well. So it's also difficult to program these. If anyone's worked with microfluidics, imagine programming a pixelated array of arbitrary 2D chemical patterns. It's not the easiest thing to do. Mechanical cues are powerful, like stretching tissues and compressing them, but they're also difficult to template and program and kind of dynamically adjust, and we don't quite know the input-output function yet. Going through other departments in engineering, you get to electrical control, and just maybe that could work. We're really good at manipulating electrical cues. Could we couple that to tissues? And if you're thinking of this, you're not wrong. There's a long and not so great history to bioelectricity. It's very interesting, but there's pretty shady past. But hidden inside a lot of the quack medicine and the strange behaviors and pop cultural aspects of bioelectricity are some experiments from the 1850s that were quite powerful and revealing. So this is Emile Dubois-Reymond, who started electrophysiology as a field, and he's famous not only for performing experiments shirtless because he thought he looked really good, but doing an experiment where he cut his thumb and measuring a naturally occurring electrochemical current in the injury site. And this is a modern reconstruction of that in both human tissue and a rat corneal tissue. And you can see that shortly after wounding, there is measurable electrochemical current. And it isn't an accident that it's there, and it isn't simply a fluke that doesn't have any impact, because it turns out that when you expose cells to this field, it can bias receptor distributions in the membrane. And if you think about receptors as little antennae that respond to different environmental cues, they all have a charge associated with them. And if you have an external DC field around the cell, it can gently pull these receptors to one side of the cell. And that concentrates them and acts like what people think is a compass signal. So now you can give a cell a forward and a backward direction. And people think this evolved for wound healing and other aspects of development because it's a useful navigational cue. And so if you inhibit it, you don't necessarily heal as effectively. So it's a natural process that guides cells and uses electricity to do so. Maybe we could use some tools to program that, was the concept. So we built a microfluidic device. We'll take a closer look at the new designs and some of the engineering challenges in a moment. But you've seen enough videos to know what these cells should look like on their own. We said, let's try to use electrical fields on large tissues to steer them kind of like sheep herding based on this process, which is called electrotaxis. Sorry, I forgot the name before. So here's that working. We turn an electric field on at about a couple volts per centimeter here. And you can see the cells transition from general kind of non-directional motion to fully directional motion. And we can make them dance back and forth by changing field polarity. It's a lot of fun stuff. So we did a bunch of work on this, and this was the last part of my PhD. And we came back recently and said, what are the actual limits of this? How much control do we really get? Can we improve the engineering to really steer these things and start to do something more useful with it? So we built a device, which we call sheepdog. It's an excellent acronym. And galvanotaxis, the G in the dog, is another word for electrotaxis. So what this device is a 2-axis stimulation system. It's an electrochemical bioreactor. It keeps cells alive at body temperature with continuous exchange of media and gas. And it has 2 axes, X and Y, for stimulating them using electrotaxis to steer them around. And that device is assembled using layer-based microfluidic fabrication, basically a lot of benchtop rapid prototyping techniques to produce a predictable microchamber that you can really simulate, engineer, and design custom tissues inside and keep them alive. And we use a lot of these cheap techniques that we like because if you don't have to go to the clean room and you can do it on the benchtop effectively, it tends to be fast and cheap and you don't have to wear the bunny suit. We use silver chloride electrodes and computer control and feedback to sort of stabilize the fields inside. But where this really needs more work is microelectrical approaches, better microfabrication of electrodes, better materials to avoid electrochemical breakdown and nasty byproducts that can kill cells. Pretty much the whole field uses silver chloride right now, or platinum in certain contexts. So we need some better solutions to make something that's less crude than just 4 slabs of silver chloride stuck on either side of a thing. But this proved the concept for us. If you have suggestions, I'm happy to listen to them because it's a wide open field. This is what it actually looks like in practice. Definitely interesting, sciencey looking device covered in technical chewing gum. And it works really well. We can steer tissues on command in any direction now. So this is a right turn in primary skin cells taken right out of the mouse. And they're moving exactly as we tell them to do. And we can alternate between X and Y at different frequencies to get cells to move at a given angle. And if we go fast enough, they experience the average field. So they can't tell that the field's changing. And basically we go faster than they sample. So we can tell them to go any direction in 2D now, just using X and Y electrodes. And that means we can make them go in a whole circle by gradually changing the field direction over time. So this is programmable tissue, and it's a material that we now have a way to steer in real time by changing the field shape. You can easily imagine you could take something like a puncture wound or a slit wound and cause it to converge maybe faster. So that's something we're looking into now, is that it's fully feedback controlled, programmable. You can do it in real time. And that means that moving forward, there's a lot of fun directions to consider. Nobody really fully understands how this process works, and understanding a little bit more about that biologically can help us design better systems that might be more efficient for the electrodes or have greater control over the cells. We also want to try it in things like 3D tissues and stem cells to try to induce controlled growth in more advanced systems than simple 2D models. And there's a ton of technological applications. So closed loop, real-time stimulation in a Band-Aid for wound healing, these microelectrode bioreactors that can do organ on a chip or ex vivo organs. So you print an organ and then maybe we stimulate it to produce some sort of physiological response inside. We're still exploring the different areas, but you can imagine that this is a neat set of approaches that definitely need some better engineering tools now that we're starting to understand just how powerful the response is for electrotaxis. So that's the first area of interactive control. And the last thing that we'll end on quickly is the inverse problem. So you can steer a population from the outside using a sheepdog, and I call that the outside-in method. You have an agent that's obviously not part of the group that manipulates it. The other way of controlling a group is to sneak into the group as if you're a member of the group and then incite rebellion or do something strange from the inside. It's like a person shouting fire in the theater deliberately to produce a certain response. For this to work, you have to produce an object that is recognized by cells in the tissue as one of their own. Now that has a lot of interesting applications again. You can produce new biomaterials that can induce new responses from the tissues that contact them. You can control growth, you can control differentiation. You can hopefully make more stable implant interfaces or improve sort of microelectronic integration for neural probes, which is something we're looking into as well. What does this look like though? And what does it mean and how do you do it? So in 5 minutes, here's the basic overview of cell-mimetic biomaterials. There's really only two kinds of interactions you need to worry about between cells and materials. One is a substrate material. That's like the floor, collagen, fibronectin, those soft biopolymer connective materials in the body that act as sort of structural support and allow cells to move around. Alternately, cells can attach to each other, and that uses adhesion proteins. And these are orthogonal mechanisms. They do not use the same protein. And that means that you have ways to target a material just by changing the proteins incorporated in that material. And what's interesting is cell-cell changes the coupling. It changes the types of collective behaviors because you can start to make an object that shakes hands with all the cells around it as if it's one of them and really integrates in and infiltrates. So there's some fun stuff you could do if we could make this work. And what's surprising is that the whole history of medical biomaterials has favored floor-based materials. Back to the earliest Western manuscript on regenerative medicine, which was called How to Make a New Nose for Someone When It's Off Entirely and the Dog Has Eaten It, which is a fantastic paper title. That paper describes methods of restructuring a missing nose using skin from your arm and microfiber cotton scaffolds, which I would love to get into more, but let's move on. We've been doing this a very long time, even much earlier than this, but these microfiber scaffolds, bone screws, fancy RGD peptides that mimic collagen, taking tissue from an organism and removing the cells from it and then putting new cells back on, that all works. Modern hydrogels and sort of structured materials that you might use in bioprinting, those all mimic the floor. They give the cells something to really latch onto and crawl around on. There aren't any materials that mimic cells right now. So when we started to do this, the question we asked was, what's the most fundamental kind of proof of concept demonstration of this? So here's our two cells holding hands again. Now here's a 3D view of that. Not only is the protein different, the feet are in green, the handshake is in red, but the geometry is different. There's a 3D aspect to handshaking in these types of tissues. And so what we did was say, well, maybe we can use that protein from handshaking and coat that on materials in the right 3D context to mimic this cell-cell adhesion. So what that looks like is you have two cells that are hanging out next to each other. This is the 3D cross-section. We're going to replace one of those cells with a fake cell, but we still want this cell to shake hands with the fake cell and trick it. And so we said, you got to have this cell recognize our material as another cell and then stick to it stably. And we think we can do that with this E-cadherin protein, which is a very common handshaking protein in cells. And so we built this using microblocks that are just cut by razor writer out of PDMS, silanized on one side, and conjugated with synthetic E-cadherin. So this is like a wall with billions of hands sticking out of it, waiting to shake your hand. And then when they shake your hand, they don't let go. So it's a little bit creepy, but the cells like it. And what we do is we have a healing tissue move up and collide with this. And we're trying to mimic healing that way. And everything's sitting on a bed of collagen. So in cross-section, you have a train of cells all shaking hands with each other and shaking hands with the wall, and they've got feet on the floor. And we built this interface just using benchtop microfab, took some images to prove that you really get this nice interface between floor and handshake mimic, and then introduced it to some cells. So here are two real cells shaking hands. Here is our cell shaking hands with a fake cell. This red protein only appears when the cell identifies another cell. Our wall here doesn't have any colors on it. So what you're seeing is entirely from this cell recognizing another cell. And you can see its feet are also in the right plane there. So then we did a wound healing assay and we let this sheet of kidney tissue heal against a cell mimetic boundary. So in 3D, you have a cross section down here and top-down view up here. And you can watch this collide with the wall and light up in red. Again, it only lights up in red when it thinks it's shaking hands with another cell. And you can see that little 3D blob right at the bottom there as it builds up against the wall. This really does work. If you block the wall with a passivation chemistry, those cells at the wall never express a protein there. And cells at the edge of a tissue that's growing and healing, they don't see anything to collide with, and they have no red protein at the boundary. You only get the handshake at a functionalized wall. And it does weird things. We thought it would really mess with tissue behaviors, but what it does instead is produce a stable interface. So if you have a wall that's inert that cells can't interact with, so a little microstructure that they can't bind to, they slide past it and it really pulls the whole tissue along. If you have a wall that's that mimics cell adhesion, they lock onto it and they don't really go anywhere. The same cell at the beginning is still there, and the rest of the tissue is kind of stuck. So we inadvertently made a very stable implant interface. And you can think of that as like a no-slip boundary from fluids. So I'll just summarize and end with this, with some exciting areas that we're looking at here. The first is that this is a whole class of materials that can be engineered into microstructures that have never been tried before. Everything on this list and more mimics different types of cell-cell interactions. So for instance, epinephrine are cell repulsive interactions. When two cells have these, they might push each other away and keep from binding. Every different class of cell has a unique cocktail of handshakes, or you can think of them as secret handshakes that they're expecting. So if you can identify those, you can mimic them. We're building neural microelectrode arrays that can provide a stronger electrical coupling and a lower contact resistance, essentially, between the cell and the electrode recording interface. And we have some interesting data there that looks like by mimicking neural cadherin handshaking, we might be able to improve stability and signals, at least in brain slices. And lastly, we're looking into making 3D microstructures down at the scale of cells that incorporate exactly the signals we want and might be under our control. So maybe light or magnetically actuated materials that you can 3D print, functionalize with something you want on the surface, and then try to get them to sneak into a tissue as if they're part of it. And they don't work very well right now. This is a control one. The surface chemistry isn't working yet, but you can see how the cells are pushing just this block of SU8 polymer around. So you can imagine you can really make some much fancier structures with modern microfabrication or printing techniques. And if you get the surface chemistry right, we can stuff those into a tissue and maybe totally change its behavior from the inside out. So, oh yeah, we also play with water bears. I'm just showing this because they're adorable. If you don't know what water bears are, they're very cute. They're vaguely invincible. They're adorable. And they're the smallest animal with legs. So we're doing a lot of work with their proteins, but we're also studying their mechanics because they're too cute not to. And this is the first sagittal video of a water bear walking side on. It's very cute. And maybe you can make mini robots about them if I have to have some sort of topical engineering connection there. Okay, so just want to thank my group for this and emphasize that it's a pretty diverse background of mechanical engineers, molecular biologists, chemical engineers, electrical engineers, a lot of great collaborators. And we are still in the process of filling up because we're just 2 years old. So if anybody's interested in joining us, let me know as well. Thank you very much for your time. And thanks, Ryan, for the invitation.
So thank you, Daniel. If we were in real life, there would be an applause, but we just have YouTube for now. So first of all, I love your graphics. Those were amazing. And we have a couple questions and also some comments. So Max said, Sean the sheep fan. And then Luke Weiss says, very cool, thanks. Related to the 2D tissue shapes, what do the boundaries look like? Specifically, over what length scale are the boundaries well defined? So I think that was in reference to like the beginning of your talk where you had the two there, yes.
The boundaries of these are rough on the cell scale. And it's sort of hard to appreciate that there, but it's not a perfectly flat boundary. And they will punch into each other a little bit and kind of push back and forth. And it turns out that whoever is denser will push the other one out of the way. Whoever's larger might push. If you have cancerous cells and non-cancerous cells, they'll push differently. But something known as boundary smoothing happens in the body when you have different zones during organ compartmentalization. And what really matters here turns out to be the fluidity of that interface. So how strongly the cells attach to each other. And then if they have any other cell proteins that manipulate that strength. So it's sort of a weird concept, but if they're all holding hands with each other, and then they also start expressing a little bit of the cell repulsive protein, it means they hold on to each other, but they kind of push away a little bit, which fluidizes the interface and allows it to relax and get flatter. So there's a lot of good stuff going on there, and we haven't really started to dive into it too much. It's a great question.
Really cool. So Ryan asks, have researchers demonstrated the ability to create photomaterials with embedded E-cadherins, or do they always have to functionalize post-fabrication?
You want to do it? E-cadherin is not cheap, but I don't see any reason why it wouldn't work. You would just waste everything on the internal volume. But if it got to the outside, that's justifiable.
And so now my question is two questions. One, can you measure cell adhesion to a surface depending on how much electrical current you have to put in to make them move? So like if you have to put in more current to make them move, does that mean cells are more adhered to a surface?
That is a great question. I don't think it translates that way because we don't know what the field does to their feet. It could be that it makes their feet pull harder or it gives them more feet. So we're not actually sure how to translate that. You can flow fluid over them really quickly and sort of strip them off the surface by shear force. And that definitely tells you how strongly they're attached. But the field is doing something a little different. So it's a fun thing to think about though.
Got it. And then my second question, have you ever thought of doing the same experiments but with biofilms, like microbes?
There are a lot of people working on biofilms here. They seem like a pretty fun system. I think there's some fun work coming out soon too on biofilm interactions. And I know like Ned Wingreen and Bonnie Bassler here have done some work on sort of how the aspect ratio of bacteria within the biofilm leads to 3D structures kind of forming as they start to wrinkle and ruffle. So they're a pretty neat material as well. And they don't die as easily as mammalian cells do, which is an advantage. And I just realized I went for 30 minutes rather than 20. Sorry about that.
That's okay. So one other comment from Luke Weiss. He says, an idea, smiley face, a self-sorting cell differentiating chip where the differentiated cells migrate to one side via design surface chemistry.
That probably works. And there's a lot of antibody-based sorting and separation based on surface adhesion and sort of rolling of cells, and that they slow down or attach on pattern surfaces. So I bet you could do that. We've wondered if you can do that with field stimulation and kind of take two different populations and split them if they have differential field responses. There's a lot of interesting things you can do once you start to tap into their native collective behaviors and manipulate them. That's a fun idea too.
Okay, well, since we are a bit over time, thank you. If you have any other questions for Daniel, feel free to email him, I think is the best way. You can also write comments on the YouTube after the talk is over as well.
You can email here.
Daniel Cohen at princeton.edu.
Yep.
And tune in next week because we have Professor Sam Amamnejad from UCLA presenting on emerging wearable bioelectronics. So thank you for tuning in, and thank you, Daniel, for presenting. That was amazing.
Yeah, thanks for inviting me.
Awesome. Thanks again. All right. Bye, everyone. Okay. And we are good.